For the remainder of today's session, we will be talking about advanced EEG methods and MEG. One advanced EEG method is the independent component analysis. Independent component analysis, or ICA, works in a way that you decompose the EEG signal into the activity of statistically independent components, so that you identify some kind of cluster that is then resembling maybe what we could call a neural ensemble. So this is basically a mathematical approach to circumvent the inverse problem, because even though we cannot solve an equation system that is underdetermined, as we've seen earlier in this session, it is still possible to approximate that with mathematical methods by looking at such statistically independent components. The analogy would be to identify the sound of a violin from an orchestra. So if you're, for example, sitting here, you get an impression, of course, of all instruments here, and to isolate the violin just by sitting here is more difficult than if you, for example, can also sit here and here and here, and then you can basically extrapolate where in the orchestra the violin is placed. Another method is classifying methods, so for example support vector machines, forward encoding models, machine learning algorithms, or dipole localization, and these classifying methods make use of the full complexity of EEG data with computationally demanding approaches and algorithms. And then another approach are brain-computer interfaces, where you can control computers or machines with EEG, and frequency-based analyses. For example, you can use specific frequencies in the EEG band to reveal patterns that are otherwise hidden in the EEG signal, because there might only be a deflection in a specific frequency due to your experimental manipulation. So let's have a look at how brain-computer interfaces work, just to give you an example. Here we use an algorithm that is trained to differentiate between different EEG patterns. And then after the training, the computer receives input, new data from the EEG system, and then can be controlled by a person who is using their thoughts to induce different EEG signals, and then as a result also induce different signals into the computer. So this can, for example, be used to help disabled people to be more independent. So how does this work in detail? This is the training procedure. This is one person, and in one trial they are instructed to move, or not to move the left arm, but just to imagine moving their left arm. In another trial they have to imagine to move their right arm. And then the data in these trials for thinking about moving the right arm or left arm, these data are then fed into the algorithm, as many trials as possible, and the algorithm needs to learn to differentiate between these two signals. How does this work? I will explain this for an example of two electrodes. So imagine this scatter plot shows the voltage in two different conditions the voltage at two different electrodes for the green and blue conditions. So, give you an example how to read this. For example, this is one trial, and here participants were instructed to thinking about moving their left arm, and the voltage at electrode 2 was somewhere here. The voltage at electrode 1 was somewhere here. This is another trial. Here, participants were instructed to think about moving their right arm, and the voltage at electrode 2 was like this. Voltage at electrode 1 was like this. So each dot here represents one trial, and the color indicates what they had to think about. Now you can maybe see a pattern here. So again, our algorithm needs to be trained in a way to tell these dots apart. So how do we do this? Maybe you want to think about this for a moment. So an obvious way to do this here would be to draw a line like this. So this tells apart the blue dots and the green dots relatively well. So there would be some classification errors, so the green dots down here and the two blue dots up here. But in principle, we could tell the green and blue dots relatively well apart. So our classifier, our algorithm would now just say, okay, anything above the red line that is coming, that's when participants are thinking about moving their left arm. Anything below the line is when they are thinking about moving their right arm. So if you get new data, you can just use this classifier to then read the participant's mind in a way that you have let's say you have a completely new data set and you have a dot here, then the algorithm will say, well, this is probably going to be, they are probably going to be moving their left arm. Then you get a dot here, a new trial here. Now they don't need to be instructed, they just think about moving their right arm. You get a dot here, the algorithm tells you, okay, below the line, so probably moving the right arm. And then you can connect this classification with the interaction with the computer. So for example, when they're thinking about moving the left arm, a dial turns around that points at different letters, and then when they think about moving their right arm, it will stop, and then you have the input to the computer. Or you can move a mouse arrow. Any kind of application is really possible. So this works relatively badly if you only have 2 electrodes. In fact, you typically take at least 64 electrodes for such brain-computer interfaces, maybe even more if possible. And of course, then this is mathematically much more complex. But the principle is the same. You need to find a red line that differentiates the different events. So this is how brain-computer interfaces work. Let's have a look at frequency-based methods. So I mentioned earlier that there's different EEG frequencies. So for example, alpha and beta have different amplitudes, different frequencies. And specific cognitive processes can be strongly linked to one frequency band alone. So the experimental manipulation, or maybe if you're looking at inter-individual differences, those may only be present in one of the frequencies, and by just averaging across all frequencies, that is, by just looking at the raw EEG data, you might not be able to identify it. But if you decompose the signal into different frequencies, then you might be able to find it. This is what I've shown you earlier with the Fourier transform. So imagine there's only a variation between 2 experimental conditions in this frequency, this red line here. Whereas this and this remains the same, then if you only look at the observed EEG signal, this might be less salient or might even be hidden. But by decomposing it, you could identify the EEG signal in that frequency and how it responds to the experimental manipulation. And this is a way to look at time frequency plots. Time frequency plots is one way to look at such experimental differences in specific frequency bands. So what this basically shows is these are 2 time frequency plots. The x-axis shows time, so this is similar to the ERP that we've talked about before. This could be a classical ERP, as you can see here. This is the onset of a stimulus, for example. And then on the y-axis, you don't see the amplitude of the ERP. The amplitude is actually color-coded here. But on the y-axis, instead, you see the frequency. So, for example, if you compare condition A and B, what you can see is that there is some kind of positive amplitude in this range, like maybe 8-10 hertz. So this is alpha band. So there's a positivity in the alpha band at around 200 milliseconds. And here in this condition you don't really see this. This is not near zero. But instead you see for slightly higher frequencies, around maybe 13, 15 hertz, you see a negativity around 400 milliseconds, which is less pronounced here. You don't really need to understand the specifics here. This is really just to illustrate how you can identify where in the time frequency spectrum two conditions may be different. In this case, this area here was a statistically most significant, identified as most significant, so that's why this is highlighted here. But this is a good way to see, okay, what time and what frequency band do we see differences. Okay, so we've talked a lot about ERPs. Let's summarize the advantages and disadvantages. So an advantage of EEG is that it has a high time resolution, so 1000 hertz or even higher. So it allows to online track cognitive processes. So as soon as something is changing in the brain, you can see it immediately in the EEG signal as well. And thus it also allows us to capture very transient processes. Another advantage is that ERPs are closely linked to specific cognitive processes. So because of decades of research, for a lot of EEG components, it is pretty clear what they indicate. And so they can be used as markers for specific cognitive processes. And then, for example, if you have a behavioral difference between two conditions, you can use ERPs to see which cognitive process is responsible for the behavioral difference that you see. It's also non-invasive, so kids stay happy if you record EEG from their scalp. And it's also easy to get ethics approval because of that. You can use it for healthy adults, for children, for patients. So a wide range of use. And the acceptance is also usually very high. One slight disadvantage in terms of the acceptance is sometimes that you have some gel remains in your hair, and that's you need to wash your hair to get rid of it. But apart from that, the acceptance is generally high. It's also cheap, so the equipment for a full lab starts around 25k pounds, and there's hardly any additional running costs apart from buying some gel and maybe cleaning some towels for participants after they wash their hair in the lab. Disadvantages of EEG or ERPs as the bad spatial resolution that we've talked about. However, there are some advanced analysis methods such as independent component analysis that allow a reasonably good spatial resolution. But this is always computationally demanding, so it's not easy to do these analyses. Another disadvantage is that you need a lot of repetitions to get a good signal to noise ratio. So this could be unsuitable for paradigms where you have, for example, surprise trials. So if you're interested in how people respond to a seldom stimulus, that would be difficult to assess because you would have to present it like 50 or 100 times, and then participants get adapted. Another disadvantage is that ERPs are sometimes particularly tricky when you plan the experimental design. So researchers that are not used to using ERPs or EEG, but behavioral studies rather, they sometimes struggle with with some confounds that might only be a problem in the EEG world, but not in behavioral experiments. So this needs careful planning, which is sometimes more complicated than for pure behavioral experiments. And another disadvantage is, of course, something that I've mentioned a couple of times before. It is always prone to noise because the EEG signal is so tiny deviations in the voltage that a lot of other things can affect the EEG signal and at times be much stronger. So it's unsuitable for very difficult for experiments that require motor activation, for example, running. Imagine you're on this treadmill. That means, first of all, you're sweating, so that might be a problem for these drifting electrodes that we've talked about. But also there's just a general noise due to the movement of the electrodes and the cables as well. But as you can see, people do try it, and people do try to cope with the noise that is induced by such conditions. So it's a problem, but there's also approaches to address this well. Okay, so far we've talked about EEG, electroencephalography. There is a related measure, which is the magnetoencephalography, or MEG. So to understand what MEG really is or where it comes from, let's go back to the dipoles. So as you remember, all EEG activity comes from postsynaptic potential, so the outside of such neurons, which is, for example here, more negative near the dendrites compared to the axon. So you have this dipole going into this direction. And you might remember this from physics class in school. Whenever you have some current that is running along one axis, you also induce automatically a magnetic field. This is perpendicular to the current that emerges, which is the Maxwell equation. So instead of current, MEG uses the magnetic field that is induced by such dipoles. Instead of event-related potentials, we have ERFs, event-related fields, because it's magnetic fields, not potentials. And the brain's magnetic field is very small. It's 10 to 1000 femtotesla, which is compared to the Earth's magnetic field. This is like 25 to 65 billion femtotesla, so you see the relation. And this already hints at a problem, because we're looking at such tiny deviations in the magnetic field that MEG requires some very demanding technical requirements. For example, you need to have superconducting sensors that are capable of detecting these magnetic fields, and you get superconducting sensors by bathing them in liquid helium, so very cold. And this is, of course, a lot of maintenance and costs that are involved here. Right, so in order to get to measure these magnetic fields, you need a very low impedance to amplify the magnetic fields and not any noise. So to compare MEG and EEG, you have the same high temporal resolution. Both are non-invasive. MEG has an excellent spatial resolution because the magnetic field passes unaffected through brain tissue and skull, so it is less blurred compared to EEG. And EEG is much cheaper than MEG, both in purchase and maintenance. So for example, for EEG you don't need the liquid helium, of course. ERFs are less clearly understood than ERPs. This is mostly due to much more experience with ERPs, so they are much better understood because there's just been so much more research on ERPs. So it's not a disadvantage per se, it's more due to the history of MEG. And then depending on the origin of the neural signal, it might be that EEG or MEG might be more likely picking up a signal. So this is explained here. Imagine you have a current running this way, and then you have the magnetic field perpendicular to that. And then imagine you have a dipole which is parallel to the skull, like here, then you would get a strong MEG signal because the magnetic field varies quite a bit, but a weak EEG signal because the current is running along the skull and cannot be measured. And this is showing you that where the EEG signal emerges, where the dipole is located, that might actually indicate which method might be more suitable. So here, when the dipole is parallel to the skull, which is shown here, then MEG would be better. Here, when the dipole is perpendicular to the skull, then that would be better for EEG, and MEG would not pick up on this very well. And this is often related to where in the cortex this is. So here, as I said, you have the dipole perpendicular to the skull. So on the main surface of the cortex, here the dipole is perpendicular to the fold of the cortex and parallel to the skull. So here MEG would be better, here EEG would be better. Of course, oftentimes these dipoles are not exactly perpendicular or parallel to the skull, but in other places, so they typically can pick up a signal with both MEG and EEG, but one or the other might be more suitable. Here's one application of MEG in medical practice, so you can use it for preoperative mapping of the somatosensory cortex. So when you have a tumor like this, in this case this is near the sulcus centralis, then you want to identify exactly where the somatosensory areas are surrounding this tumor, what they're responsible for, so you can spare those areas which are considered most crucial for maintaining a high life quality. So you do this by using some kind of tactile simulation, finger, arm or leg, then record the ERFs, that's this one here, and then you use inverse modeling of dipoles, again addressing the inverse problem, and then from the overall signal you can kind of see what's the most likely origin of the tumor. And then as a result you can calculate dipoles for these sensory areas, so these red dots here, toes and fingers, and this can be compared to the location of the tumor. And then if you can try to find those areas which are least where cutting those areas out would be least affecting the life quality. So much about EEG and MEG. Thank you very much for your attention, and see you next week when we will be talking about fMRI and lesion studies.